Every year, across villages, suburbs, and rural homes, the same quiet ritual repeats itself. A fire burns through the evening. A wood stove warms the kitchen before sunrise. A backyard fire pit glows for a few hours and fades into embers. And when the fire is gone, what remains is always the same: a thin layer of gray powder. Most people treat it as waste. They sweep it into a bucket and dump it somewhere along the edge of the yard. Some scatter a little in the garden because they once heard that ash might help plants. Others avoid it completely, worried that it could damage the soil, so the ash disappears quietly. But inside that powder lies something far more interesting than most gardeners realize. Because wood ash is not simply the remains of a fire. It is the concentrated mineral memory of the entire tree that burns to create it. Every element that once moved through that tree's roots and branches remains there in concentrated form: calcium, potassium, magnesium, trace minerals that plants rely on for root development, leaf formation, and flowering. For centuries, farmers understood this instinctively. Long before modern fertilizers existed, wood ash was carefully collected and returned to the soil. Entire industries even grew around it. During the 18th and early 19th centuries, barrels of ash were traded across Europe and North America because they could be processed into potash, one of the most valuable mineral fertilizers of the time. In other words, the material most people sweep into the trash today was once valuable enough to ship across oceans. So, why did something so useful slowly disappear from everyday farming? The answer begins with a problem every experienced gardener eventually encounters. Raw wood ash can be surprisingly harsh. At first glance, it looks harmless, almost like flour or chalk powder. But chemically, fresh ash behaves very differently from soil. When it mixes with water, many of its minerals dissolve rapidly, creating a strongly alkaline solution. In simple terms, the pH rises quickly. Most garden soils exist in a delicate balance between slightly acidic and neutral conditions. In that range, plant roots and soil microbes cooperate to regulate how nutrients move through the ecosystem. Raw ash disrupts that balance. When applied too heavily, it can push soil chemistry upwards so quickly that roots struggle to absorb nutrients and microbial communities decline. Leaves begin to yellow. Growth slows. Roots struggle to expand. Many gardeners experience this once and conclude that ash is simply harmful. But that conclusion misses something important. The problem is not the minerals inside the ash. The problem is the way they are delivered. In natural ecosystems, minerals almost never appear suddenly. In forests, nutrients move through slow cycles of decay and transformation. Leaves fall. Fungi digest them. Microbes reshape their chemistry. And only then do those nutrients return to the soil in forms plants can safely absorb. Raw ash skips that entire process. It releases concentrated minerals instantly without the biological buffering that normally moderates them. And that is where the real story begins. Because some farmers eventually asked a simple question. What if ash did not go directly into the soil? What if it passed through a transformation first? One that allowed microbes to soften its chemistry before plants ever encountered it. Instead of scattering ash onto the ground, they began mixing small amounts with water and natural sugars. Then they waited. Within a few days, something unusual began to happen. Tiny bubbles formed on the surface. The sharp mineral smell softened into something slightly sour and earthy. Invisible microbial communities awakened inside the liquid. What began as gray dust slowly turned into a living brew. When that fermented mixture was diluted and applied to plants, growers noticed something surprising. Instead of the shock caused by raw ash, plants responded more gently. Leaves deepened in color. Roots expanded faster. Flowering sometimes arrived earlier. The change was subtle at first, but consistent enough that more gardeners began experimenting with the idea. Some added only a spoonful to irrigation water. Others diluted roughly 100 milliliters of fermented ash extract into watering cans. And the results continued to appear. which raises an interesting question. If such a small amount of transformed ash can influence plant growth so noticeably, why do so few people talk about it? To understand that, we have to travel back to a time when farmers were forced to rebuild their soils with almost nothing except the materials they already had. Because the rediscovery of fermented mineral inputs did not begin in a laboratory. It began in the fields of a country rebuilding after war. To understand how fermented ash reappeared in modern gardening, we need to travel to a very different place and time. In the years following the Korean War, much of South Korea's countryside faced a difficult agricultural reality. Farms had been damaged, resources were scarce, and imported chemical fertilizers were becoming the dominant solution promoted by agricultural authorities. Farmers were told that productivity depended on manufactured inputs, standardized bags of nutrients produced in distant factories. For many growers, this created a problem. Fertilizers had to be purchased every season, prices fluctuated, and soils that had supported crops for generations began showing signs of fatigue when managed purely through chemical inputs. Among the farmers who began questioning this direction was Cho Han-kyu, who would later become known for developing what is now called Korean natural farming. Cho was not searching for a miracle product. What interested him was something much simpler. How had traditional farming systems maintained fertile soil long before industrial fertilizers existed? Instead of focusing on synthetic nutrients, he began studying biological processes already occurring in nature. In forests, fallen leaves decomposed through microbial activity. Animal manure fermented naturally before becoming part of the soil. Even plant residues left in moist environments slowly transformed through microbial metabolism into stable organic matter. Gradually, a pattern became clear. Fermentation was not just a way to preserve food or produce alcohol. It was one of nature's fundamental methods for transforming raw materials into nutrients that living systems could actually use. In other words, fermentation acts as a biological bridge between complex materials and accessible nutrients. This insight shaped the philosophy of Korean natural farming. Instead of applying raw materials directly to the soil, Cho began experimenting with ways to pre-transform those materials through controlled fermentation, allowing microbial communities to soften their chemistry before plants encountered them. At first, these experiments focused on plant extracts, compost liquids, and microbial cultures collected from forest soils. But over time, farmers began experimenting with mineral materials as well. And wood ash quickly attracted attention. Ash had always been recognized as a concentrated mineral residue. It contained potassium, calcium, magnesium, and trace elements essential for plant growth. The challenge was its harsh chemistry when applied directly to soil. Fermentation offered a possible solution. When small amounts of ash were mixed with water and a carbohydrate source such as sugar or molasses, the mixture began to ferment. Ash itself contains no living organisms, but the surrounding environment is full of microscopic life. Yeasts and bacteria exist in the air, on plant surfaces, and even on the containers used to mix the solution. When sugar enters the mixture, these microorganisms suddenly gain access to energy. Within hours, their populations begin expanding. Within days, they begin reshaping the chemistry of the liquid. As microbes metabolize sugars, they produce organic acids such as lactic acid and acetic acid, compounds commonly generated during natural fermentation. These acids interact with minerals dissolved from the ash. Instead of releasing in a single harsh burst, mineral ions gradually associate with organic molecules produced by microbial metabolism. Chemists describe this interaction as chelation, a process in which organic compounds bind to mineral ions and stabilize them in solution. The practical effect is easier to understand than the chemistry. The minerals become less aggressive. Their behavior softens, and the liquid moves closer to the chemical conditions that soil microbes and plant roots can tolerate. At the same time, fermentation introduces another important element: living microorganisms. When the fermented liquid is diluted and applied to soil, Those microbes interact with existing microbial communities in the rhizosphere surrounding plant roots. They contribute enzymes, organic metabolites, and biochemical signals that influence how nutrients move through the soil ecosystem. The result is not simply mineral water. It is a biologically active mixture where minerals, organic acids, and microbial life interact together. For farmers experimenting with this process, the difference became noticeable. Where raw ash often shocked the soil system, fermented ash behaved more gently. Instead of overwhelming roots with sudden alkalinity, it introduced minerals within a biologically buffered environment. Plants seemed able to integrate those nutrients more smoothly. This did not mean fermented ash replaced every other soil management practice. Healthy soils still required organic matter, microbial diversity, water, and time. But fermentation appeared to solve one key problem. It transformed a harsh mineral powder into something living soil systems could handle. And once growers began seeing that difference, they started experimenting further. But to understand why plants respond the way they do, we need to look deeper. Not at the soil itself, but at what happens inside the plant once those minerals begin moving through its tissues. When gardeners describe plants suddenly coming alive, they usually talk about what they can see above the soil surface. Leaves deepen in color, stems become thicker, new shoots appear, and flowers begin forming sooner than expected. But the real transformation rarely begins in the leaves. It begins at the root. Plant roots are not passive tubes absorbing whatever nutrients happen to be nearby. They are part of a highly active biological environment known as the rhizosphere, a narrow zone of soil where plant roots, microorganisms, minerals, and organic compounds interact continuously. Inside this zone, nutrients rarely travel directly from soil into plant tissue. Instead, they move through a series of biological exchanges. Microbes dissolve minerals. Organic acids reshape their chemical form. Root hairs release sugars that feed microorganisms, which in turn make nutrients available to the plant. It is a complex negotiation rather than a simple transfer. When raw wood ash enters this environment, that negotiation can collapse. Because raw ash dissolves rapidly, it can push soil alkalinity upward very quickly. In those conditions, some nutrients become less available, while others accumulate in forms that roots cannot safely absorb. Microbial communities may decline, weakening the biological network that normally regulates nutrient exchange. Fermented ash behaves differently. Because fermentation has already softened the mineral chemistry and introduced organic acids into the mixture, the solution reaches the soil in a far more moderated state. Minerals arrive gradually within a biologically active environment that soil organisms can integrate rather than resist. Once those minerals begin entering the plant, three elements play particularly important roles. The first is calcium. Calcium is often discussed in relation to soil pH, but inside plants, it serves a very different function. It stabilizes cell walls and strengthens the structural framework that allows plant tissues to expand without collapsing. When calcium becomes available in balanced amounts, root growth often responds first. New root hairs form more quickly, allowing the plant to explore a larger volume of soil in search of water and nutrients. Stronger roots create a positive feedback loop. The plant absorbs more resources, which supports further growth above the surface. The second key element is magnesium. Magnesium sits at the center of the chlorophyll molecule, the pigment responsible for capturing light during photosynthesis. Without magnesium, the plant's ability to convert sunlight into chemical energy declines rapidly. This is why magnesium deficiency often appears as pale or yellowing leaves. When magnesium becomes available again, chlorophyll production increases, and leaves regain the deep green color associated with healthy plant growth. The third essential element is potassium. Potassium acts as a regulator of movement within the plant. It controls how water flows through cells, how stomata open and close on leaf surfaces, and how sugars produced during photosynthesis travel from leaves to roots, flowers, and developing fruits. In simple terms, potassium helps distribute energy throughout the plant. When potassium levels are balanced, Sugars produced in the leaves can move efficiently toward growing tissues, fueling expansion across the entire organism. When these three minerals operate together, calcium strengthening structure, magnesium powering photosynthesis, and potassium regulating internal transport, the plant's metabolic system becomes far more balanced, and that balance often becomes visible surprisingly quickly. Gardeners may notice deeper leaf color, stronger stems, or earlier bud formation. These changes are not the result of a stimulant forcing plants to grow faster. Instead, they reflect the restoration of internal processes that were previously limited by mineral imbalance. Plants do not require enormous quantities of many nutrients. What they require is the right balance at the right moment. But minerals alone do not fully explain the response many growers observe when using fermented inputs. Because fermentation introduces something else into the soil, microbial life. When fermented solutions are applied to soil, they carry populations of bacteria and yeast, along with the organic compounds those organisms produced during fermentation. These microbes interact with the existing soil ecosystem, contributing enzymes and biochemical signals that influence how nutrients move through the soil matrix. Microorganisms can dissolve mineral particles that would otherwise remain locked in the soil. They produce organic compounds that help nutrients remain soluble, making them easier for plant roots to absorb. In other words, fermentation does not simply change the minerals themselves. It changes the context in which those minerals enter the soil ecosystem. Instead of arriving as isolated chemical compounds, they arrive as part of a living biological system. And for plants that evolved within complex microbial environments, that difference matters. Yet even with these advantages, fermented ash remains largely unknown outside specialized gardening communities. Few agricultural textbooks mention it. Commercial fertilizers rarely acknowledge it. And most gardeners still sweep wood ash into a pile without considering what it might become through transformation. Which raises a larger question. If wood ash once played such an important role in agriculture, and if fermentation can reshape its behavior so dramatically, why did this knowledge fade from mainstream farming in the first place? To answer that, we need to look beyond soil chemistry and plant biology. We need to look at the historical transformation of agriculture itself. Long before gardeners debated whether wood ash belonged in the compost pile or the trash bin, it held a very different status in agriculture. For centuries, ash was not considered waste at all. In many regions, it was a valuable raw material that farmers collected and traded alongside their crops. When forests were cleared for farmland across Europe and North America, enormous quantities of timber were burned. What remained after those fires, barrels of pale gray ash, was carefully saved rather than discarded. The reason was simple. Ash could be converted into potash, one of the earliest mineral fertilizers used in large-scale agriculture. The word itself reveals the process. Potash literally means potash, referring to the traditional method of soaking wood ash in water and evaporating the liquid in large iron pots. The resulting crystals contained concentrated potassium salts, which became essential ingredients for farming, soap making, glass production, and textile processing. During the 18th and early 19th centuries, entire rural economies developed around this material. Farmers near forested regions often produced potash as a secondary product. Barrels of ash were transported by wagon or riverboat to industrial centers, where they were refined and sold throughout expanding agricultural markets. In many places, potash was traded almost like a crop. Newspapers listed its price alongside wheat and cotton. Merchants shipped it across oceans. Governments even encouraged production because it supported multiple industries at once. In other words, the gray powder left behind after a fire once played a central role in the economic life of entire regions. But this system began to change during the late 19th and early 20th centuries. Chemists discovered large underground deposits of potassium-rich minerals that could be mined directly from the earth. At the same time, industrial chemistry advanced rapidly, allowing manufacturers to produce synthetic fertilizers containing carefully measured combinations of nitrogen, phosphorus, and potassium. These new fertilizers had several advantages that traditional ash could not match. They were uniform. They could be produced in enormous quantities. And they could be stored for long periods without changing their composition. Agriculture was entering an era of scale. Large farms required inputs that could be delivered predictably across thousands of hectares. And standardized fertilizers fit that system perfectly. Wood ash did not. Its composition varied depending on the species of tree that had been burned. Hardwood ash contained different mineral ratios than softwood ash. Some ash included charcoal fragments or other residues that complicated chemical analysis. From the perspective of industrial agriculture, this variability made ash difficult to standardize. And when farmers applied raw ash without understanding its chemistry, the results could indeed be problematic. High alkalinity sometimes disrupted soil balance, reinforcing the warnings that agricultural advisors began repeating throughout the 20th century. Gradually, ash lost its reputation as a useful soil amendment and became associated with risk instead. As chemical fertilizers expanded across global agriculture, the everyday knowledge surrounding ash slowly faded from practice. The material remained familiar in fireplaces and stoves, but its connection to soil fertility became increasingly obscure. Yet while mainstream agriculture moved toward industrial inputs, a different movement was quietly forming elsewhere. Small groups of farmers, researchers, and gardeners began looking again at older agricultural practices. Not out of nostalgia, but out of curiosity. They were asking a simple question. Could some traditional methods still hold value if they were understood through the lens of modern soil science? Instead of discarding materials like wood ash entirely, they began experimenting with ways to integrate them more carefully into living soil systems. Fermentation became one of the tools that made this possible. Because fermentation allowed harsh mineral materials to be transformed biologically before entering the soil, it reopened the possibility of using substances that had once been considered too aggressive in their raw form. Wood ash, rich in minerals but chemically intense, suddenly looked different when viewed through this biological perspective. It was no longer simply a powder to scatter on the ground. It became a material that could pass through microbial transformation before returning to the soil. And once that transformation occurred, growers began noticing something interesting. The ash behaved very differently. But to understand why this rediscovery is happening now, and why techniques like fermented ash are attracting attention again, we need to look at something deeper than history. We need to look at the changing way scientists and farmers are beginning to understand soil itself. During most of the 20th century, agriculture moved steadily toward a single goal: scale. Fields became larger, machinery became more powerful, and fertilizers were engineered to deliver nutrients in exact quantities across vast areas of farmland. The system was remarkably effective at increasing crop production, and for many decades it helped feed a rapidly growing global population. But this transformation also changed how soil itself was understood. In traditional farming systems, soil had always been treated as a living environment. Fertility was built slowly through compost, plant residues, animal manure, and the natural decomposition cycles that returned nutrients to the ground year after year. Industrial agriculture simplified that complexity. Instead of relying on slow biological cycles, it focused on delivering nutrients directly to plants in forms they could absorb immediately. In many situations, this worked extremely well, especially when combined with irrigation, crop protection chemicals, and modern crop genetics. Yet over time, another pattern began to appear. In many agricultural regions, long-term studies started reporting gradual declines in soil organic matter, reduced microbial diversity, and increasing dependence on external inputs simply to maintain stable yields. These changes did not occur everywhere, and they were not caused by a single factor. But they revealed something important. Soil fertility had never been purely chemical. It was biological. The nutrients plants require, calcium, potassium, magnesium, phosphorus, rarely move directly from mineral particles into plant roots. Instead, They pass through an enormous biological network that scientists are only beginning to understand fully. Bacteria dissolve minerals and release enzymes. Fungi extend microscopic filaments through the soil, transporting nutrients across distances that roots can't reach alone. Roots themselves release sugars into the surrounding soil, feeding microorganisms that in turn make nutrients available in exchange. Within this underground ecosystem, nutrients circulate through living processes rather than appearing suddenly in concentrated doses. When this biological network is healthy, soil becomes remarkably resilient. Organic matter accumulates. Water infiltrates more easily. Nutrients remain within the ecosystem rather than washing away after rainfall. Plants develop deeper root systems and become more resistant to environmental stress. When this network weakens, soil becomes far more dependent on outside inputs, and this realization has led many farmers and researchers to rethink how nutrients should enter the soil in the first place. Instead of focusing only on the chemical composition of fertilizers, they are increasingly asking another question. How can nutrients be introduced in ways that cooperate with soil biology rather than bypassing it? This question lies at the center of what is often called regenerative agriculture. Practices such as cover cropping, reduced tillage, compost application, and microbial inoculation are all designed to strengthen the biological systems that regulate soil fertility naturally. Fermentation-based inputs fit naturally into this philosophy. Because fermentation cultivates microbial activity in a controlled environment, it allows certain materials to be biologically transformed before they ever reach the soil. Organic residues, plant extracts, and even mineral powders can pass through microbial metabolism, producing organic acids and enzymes that change how those materials interact with soil ecosystems. In this context, fermented ash becomes easier to understand. The ash itself is simply a mineral source. Its potassium, calcium, and trace elements are already valuable nutrients. The challenge lies in how abruptly those minerals enter the soil when ash is applied directly. Fermentation alters that pathway. When microbes metabolize sugars in the presence of ash, they produce organic acids that interact with mineral ions and stabilize the solution. Instead of a harsh alkaline powder, the mixture becomes a biologically buffered liquid where minerals exist alongside microbial metabolites. Once diluted and introduced into soil, this mixture behaves differently. Microorganisms incorporate the minerals into their own metabolic cycles. Organic acids help keep certain nutrients soluble. Plant roots encounter a nutrient environment that resembles the gradual mineral release found in natural ecosystems. For gardeners and small farmers experimenting with the method, the preparation itself is remarkably simple. A container, water, a small quantity of clean wood ash, and a carbohydrate source such as sugar or molasses to fuel microbial activity. The mixture ferments for several days while microorganisms multiply and reshape the liquid chemistry. Bubbles appear as gases are released, and the smell gradually shifts from sharp and mineral to slightly sour, an indication that fermentation is underway. Afterward, the liquid is diluted heavily with water before being applied to soil or irrigation systems. The quantities involved are typically very small. Often only a few spoonfuls of fermented mineral extract are mixed into several liters of water. Because the goal is not to apply large doses of nutrients, the goal is to introduce minerals through biological processes that the soil ecosystem can integrate gradually. When used thoughtfully, fermentation-based mineral inputs reconnect agriculture with the same biological pathways that have regulated nutrient cycles in natural ecosystems for millions of years. And in a time when soil health is becoming an increasingly important concern, that reconnection is attracting renewed attention. Which brings us to the final part of the story. Because the real significance of fermented ash is not simply the mixture itself, it is what the mixture reveals about how soil fertility actually works. For all its potential benefits, fermented ash is not a miracle solution. Like any soil input, it must be used with care and with an understanding of local conditions. The ash itself still contains alkaline minerals, and fermentation softens their behavior but does not eliminate their chemical power entirely. This is why experienced growers almost always apply fermented ash in very small, diluted quantities. The goal is not to deliver a concentrated fertilizer dose. The goal is to introduce minerals gently into the soil ecosystem so that microorganisms and plant roots can integrate them gradually. The source of the ash also matters. Only ash produced from clean, untreated wood should ever be used in soil. Ash from painted lumber, construction waste, or treated materials can contain contaminants that should never enter a garden environment. Even with clean ash, observation remains essential. Different soils respond differently depending on their existing chemistry. Highly alkaline soils may not benefit from additional ash-derived minerals at all, while slightly acidic soils often respond more positively. In other words, fermented ash works best when it is treated as one small tool among many, rather than as a universal solution. Healthy soil still depends on organic matter, microbial diversity, water, and time. Compost, mulch, plant residues, and living roots all contribute to the gradual rebuilding of soil structure. Fermented mineral inputs simply interact with that process, helping certain nutrients circulate more smoothly through the biological networks beneath the surface. Seen in this context, fermentation is less about the ash itself and more about the pathway through which nutrients enter the ecosystem. Nature rarely delivers minerals suddenly. In forests and natural landscapes, nutrients move through slow cycles of decay and microbial transformation. Leaves fall. Fungi digest them. Microorganisms reshape their chemistry, and only then do those elements return to the soil where plant roots can absorb them safely. Fermentation recreates a small version of that process. It allows microbes to interact with raw materials before those materials reach the soil, softening their chemistry and weaving them into the biological rhythms that already exist beneath the ground. For gardeners who experiment with these ideas, the results often appear gradually. The soil becomes darker and easier to work. Water infiltrates more easily after rainfall. Earthworms return to areas where the ground once felt compacted and lifeless. These changes do not come from a single ingredient. They emerge from the slow rebuilding of relationships between minerals, microbes, organic matter, and plant roots. And that realization leads back to the simple image with which this story began. A small container of ash beside a fireplace. At first glance, it appears to be nothing more than the final trace of a fire that has already burned out. But within that powder remains the mineral memory of the tree that once stood in the forest. Calcium that strengthened its wood. Potassium that regulated the movement of water through its cells. Magnesium that helped its leaves capture sunlight and transform it into energy. Fire removed the organic structure of the tree, but it did not erase the minerals that once sustained its life. For generations, farmers understood this connection without needing to explain it in scientific terms. The ash from household fires was returned to the land that produced the wood, quietly closing a cycle that linked forests, farms, and human settlements. Over time, modern agriculture developed faster and more powerful ways to deliver nutrients to crops. Many of those tools remain essential today, and they continue to support food production on a global scale. Yet alongside those technologies, there is growing recognition that soil itself is far more than an inert medium waiting for chemical correction. It is a living system, a network of organisms and minerals interacting continuously beneath our feet. Fermented ash does not replace modern agriculture, and it does not claim to transform soil overnight. What it offers instead is a reminder. Even the simplest materials can participate in the complex biological cycles that sustain plant life when they are introduced through living processes rather than abrupt chemical ones. And sometimes the most interesting discoveries are not entirely new ideas at all. They are old practices seen again through a deeper understanding of the systems that have always supported life in the soil. So the next time a fire burns down to its last embers and a thin layer of ash settles at the bottom of the stove, it may be worth looking at that gray powder a little differently. Because what appears to be the end of a fire may still be the beginning of another cycle.